Good afternoon, and welcome to the webinar on AI for digital brain health. My name is Aureli Soria-Frisch. I'm the director of the neuroscience business unit at Starlab. Starlab is a private research and development company based in Barcelona. It's a technology transfer company turning scientific results into meaningful and useful products and services. We provide this webinar together with Neuroelectrics, which is a digital brain health company, working mostly on EEG analysis as well as on the employment of transcranial current stimulation for the treatment of different neurological and mental health diseases. We share basically a passion for the electrical brain. So the fact that information and stimuli coded information is transferred from one area to the other in the brain in form of electricity. And this gives the opportunity both to record, to read this electrical activity with neuroelectrics devices, as well as to apply an electrical field to modify this electricity within the brain for treatments. And we are going to present both the usage of AI both for interpreting this electrical brain activity as well as for modifying in form of treatments in transcranial electrical stimulation. I will start with the first part, which is focus on the usage of artificial intelligence for diagnosis, prognosis, and patient stratification. So basically, if you look at brain health, besides its large prevalence among the world population, which accounts for 20% of people suffering from either a mental health disease or a neurological disease over a lifetime, one feature that characterizes all of them is that the diagnosis is based on the subjective, or mainly on the subjective evaluation of symptoms. Besides this, the development of treatments for CNS diseases is the riskiest one among all pharmaceutical domains. So it has the lowest chance of approval, the largest development phases, and accordingly, the largest cost of development of treatments. If you see, for instance, the amount of drugs in psychiatry that were certified by the FDA between 2017 and 2019, you can see that the amount for psychiatry for mental health diseases is the lowest one. At the same time, there is an increasing prevalence of this kind of diseases over the both in high-income countries as well as in low and medium-income countries. And basically, this, for instance, bring recently the magazine Forbes to account for the usage of neurotech in the development of new treatments. And that's basically what we are doing at Starlab. We have a strong program on biomarker discovery for mental health as well as for neurological diseases. This is combined with a growth of healthcare data, which has suffered in the recent years an exponential growth, as you can see here, from the years 2013 to 2020. This has exponentially increased. This accounts for genetic data, for neuroimaging data, for electronic health records. All this data is at your hands in order to achieve better diagnostic and better treatments, normally in form of personalized treatments of mental health and neurological diseases. And this is why the purpose of the application of artificial intelligence at Starlabs. And I would present different cases of using this EEG in combination with artificial intelligence for compiling so-called digital biomarkers that allow to play different roles in the process of mental health. If you take this paper in 2017, what is the role of biomarkers in mental health and in neurological diseases? Basically, you can use them for three purposes: for the assessment of risk or the prognosis of different types of diseases, for executing or facilitating or supporting the diagnosis of particular diseases, and also for the prediction of treatment outcome. And I will present three different use cases of how can you use EEG and the digital biomarkers that you can extract with artificial intelligence for these three different purposes. The first one is focused on the use case of Parkinson's, where we have developed in the recent years decision support system. As you may know, Parkinson's has its clinical phase where all the symptoms are known, so like the tremor or the bradykinesia, but the disease starts several years before with different symptoms, among which you can find REM behavior disorder, which is a sleep disorder that makes that people that are completely asleep act during their dreaming phase. This is a disease that appears at 60 years old, and basically is a predictive risk of getting Parkinson's disease or other types of dementia, like dementia of Lewy bodies, in 30% of the cases in each of these two cases. And for this purpose, we have used EEG acquired at the phase where subjects were suffering from REM behavior disorder, apply a machine learning system in order to predict this potential neurodevelopment, neurodegeneration into Parkinson's disease or dementia with Lewy bodies. And this is the outcome of the machine learning system. Basically, you can use the outcome of these algorithms as you would use another biomarker score, and you can establish different probability distribution on these scores, and you can find out the effect size, which in our case, for the discrimination between those subjects that remain RBD from those subjects that will develop Parkinson's disease or dementia with Lewy bodies 8 years later, had an effect size of 0.75. Another application, so this same system was used as a decision support system in the Hospital São Paulo for distinguishing Parkinson's disease patients with cognitive decline from those patients that do not suffer from cognitive decline. And we have tested this decision support system in an operational environment so far. You can use this as well, this kind of artificial intelligence based biomarker in order to stratify your population between the early patients and the advanced Parkinson patients, and therefore to have a more homogeneous cohort in your clinical trial. In another type of application, we have used artificial intelligence for diagnosing attention deficit disorder. And for this, as you may know, in this disease, there is a large misdiagnosis rate, approximately estimated as a 22% of misdiagnosis rate. And we think that artificial intelligence in combination with EEG can be used for helping in lowering these misdiagnosis rates. And therefore, we have implemented a system, an artificial intelligence system that can be used in the diagnosis with high discrimination power between ADHD subjects and healthy controls. In the third use case, I would like to show you an application of artificial intelligence in the stratification of transcranial electrical stimulation response. Basically, you have in your trial a very unhomogeneous population that with the help with artificial intelligence, you can manage to split in more homogeneous group with the idea that each of these homogeneous group present a different response to the treatment. And this is what we have done. So this is our population. Each of the points is one subject, and we can split this apparently population that was homogeneous in 4 different groups. And precisely one of these, 2 of these groups present an atypical EEG response. And concretely, one of these groups with this atypical EEG response can be used to show and to predict the behavioral response when applying transcranial electrical stimulation. So only in this particular group, you can observe a statistically significant difference between the application of transcranial current stimulation. And this has been published recently in a paper in Scientific Reports. And with this, I would like to close my part, thanking everybody of the team which has contributed to the work, and thanking all the audience for the attention. We will be collecting questions at the end of the webinar, and then we will run them over the 3 presenters of the webinar. And now I would like to introduce you José Sanchez-Todo, which will present you basically the application of artificial intelligence for transcranial, for the personalization of transcranial electrical stimulation.
Hello, everyone. Thanks, Aureli, for the introduction. So as Aureli said, I'm José Sanchez. I'm the Director of Research and Development here at Neuroelectrics. And by my side, I have Nacho Moragues, which is the Customer Support Engineer that will do the next presentation right after me. And as Aureli was saying before, what we share is our passion to understand the electric brain together with a startup. And for that, we build these devices that are able to record this electrical activity with the electroencephalography, but also to inject some of the activity in order to modulate the electricity in the brain. Why do we do that? Our motivation is to treat patients. Our main focuses are epilepsy, depression, and Alzheimer's disease. And today I'm going to show you how we are planning to treat these patients in the specific case of epilepsy. So epilepsy is a neurological disease that is currently treated with antiepileptic drugs, but 1/3 of this population do not react to this drug. So the solution for them, for some of them, actually 20% of these patients, is to go to surgery. There is also the caveat that 40% of the surgery also fails. So that leads to almost 19 million people worldwide without any current treatment. And for that, we look for alternatives such as transcranial electrical stimulation. How does this technique work? As I said, it injects electricity to the brain, very low electrical currents, and this is through the scalp, so it's non-invasive. It's non-invasive treatment that can have acute and long-lasting effects, depending on the intensity, on where the electrodes are placed, and the duration of the treatment as well. And it can be focal, so it can target a specific focus, or a whole network in the brain. So this current can have different waveforms. It can have a direct current injected through the scalp that, depending on the polarity, can excite the neurons that are affected by these electric fields, or can inhibit the neurons below. Why do we need each of these cases? For example, in depression, which is on one of our clinical focus, all the patients have part of the breast activity in the dorsolateral prefrontal cortex, so we want to excite the brain. So for that, we would apply this anodal stimulation. That is this direct current. In the case of epilepsy, you have an electrical discharge that is very profound in one of multiple areas of the brain. So you want to inhibit the electrical activity below the electrode. So for that, you use the cathodal electrical stimulation. We also can apply many other waveforms through the scalp, such as alternating current at different frequencies. And for that, why do we use that in the case of Alzheimer's, for example, is because it has been seen that there are some frequencies in this electrical activity that are missing or depressed in the case of Alzheimer's disease. So we want to entrain these frequencies that are missing externally using these currents that we are applying. And for that, we make the neurons oscillate at the frequency that they have lost. So this is a bit the technique, the transcranial electrical stimulation. And as I said, we can target different networks. And this is very important because every patient is different and every patient can have different symptoms, and every subnetwork can define different symptoms as well. And this is very good as opposed to drugs, because the drugs actually target the entire brain with the different receptors that the drug actually targets. But with the transcranial stimulation, we can actually target specific areas of the brain, not the full brain. Okay, but also each patient is different, so how we can optimize the stimulation of this case? It's not just enough to place electrodes randomly in the scalp and hope that something happens. It's very important how do you place these electrodes and which intensity you put into these electrodes. And for that, we use neurotwins, which is a very fancy name for computational models of the individual brain. So the whole idea is like we have patient biomarkers, as Aureli was saying, and then we ingest these biomarkers and the data from the patient into one model of the patient, of the brain of the patient, to represent the pathology and to represent the same biomarkers as we observe in this individual. Then what we do is to optimize the stimulation virtually. So we just play with the neurotwin of the patient so that we can find the best solution for each specific person. And then we go back to the real patient and apply the stimulation protocol. Let me put you an example on how this looks like for one of the pipelines that we have. A ver si me funciona esto. Okay, yeah, okay. So we have the data from the patient in epilepsy, for example. The doctor defines the specific target where the focus is, basically, and for that it needs the MRI. So the doctor comes here and paints, for example, the left prefrontal cortex, which is where suppose the seizure starts, basically. And we inject this data in our pipelines. We use artificial intelligence to segment the magnetic resonance and to build these FEM models that you see on the screen. And with information of the target that the doctor has also given to us, we optimize this stimulation virtually, and then we go back and apply it to the patient. So this is how it looks like for one of our pipelines that just uses the information of the magnetic resonance and also the target definition from the doctor. But now let me show you a more sophisticated pipeline on how this can go, not just using the magnetic resonance of the patient, but more data as well to make it a bit more complex. So imagine we have our patient here, Paul, that is 23 years old, and it has this drug-resistant focal epilepsy. So the drugs do not react on this patient. So the doctor takes the MRI, so we can build this first version of the neurotwin, which is this geometric information of the patient, but also takes the computed tomography, for example, to know where the electrodes are located. These electrodes are invasive, and these are done for regular diagnostics of complex patients of epilepsy. So they put the electrodes inside the brain. And in order to know where these electrodes are, you need this information. And with these electrodes, you can record the electrical activity inside the brain so that you can know where the focus or the epileptic focus starts. As you see on the screen, for example, there is a focus, which is this red dot, that has a high epileptogenicity level in the temporal lobe, let's say, and then it spreads to other regions that are the orange ones, and there are some that are not affected by this seizure, which are the green ones, for example. So this is the information that we get from this SEEG data, which are these electrodes that we can put inside the brain. But another data that we can ingest in our neurotwins as well is how these areas are actually connected. So we have this diffusion weighted MRI, which is like a map of the individual brain, so that if a seizure starts in Plaza Catalunya, it will spread easily to different areas of the brain, rather than, for example, it starts in the airport of Barcelona, it should spread in a few different areas of the brain, right? So this information, this map, is very important for us to know. But how then we use it? So we have this map of the brain, and we have the different areas of the brain, so that we put in these different areas a model that replicates the electrical activity of the brain. So we can replicate healthy areas of the brain, but also we can touch this excitation inhibition balance of the brain and in the models as well to represent the different areas and also the epileptic focus. Okay, so once we have this network, this whole brain model, we personalize the parameters so that we can replicate the patient biomarker, which is the patient seizure that you see on the left, inside the neurotwin, which is what you see in the right. And this is done by a genetic algorithm, basically, that touches the parameter of the model so that we can have the patient biomarker also in the neurotwin. Once we have this personalized neurotwin, what we do is just optimize this stimulation. So we play around with the electrodes and see where we can place them so that we can reduce this epileptogenicity level in the neurotwin of this patient. Once we find the optimal solution, we go back, as I said before, to the patient. And this from here is what Nacho will show in the next presentation. So once we have the solution for each patient, what we do next. So Nacho will show us in the following presentation. And just to conclude, I'm going to outline a bit of all the clinical trials that we are doing right now with these pipelines and these and the devices that I just showed you. So we got some initial results in an open label pilot study with the first pipeline that I showed you, just using the MRI data of the patient, and we could reduce the seizure of the patient by a median of 47%. So this is very good news. But we want to improve these numbers, so we want to get further. And that's why we develop this more complex pipeline when we just inject more data from the patient and see if we can get better results. And we will start the clinical trial with these more sophisticated neurotwins of the patients in epilepsy in June this year. Another completely different line is what we do for Alzheimer's disease, which we use just the morphometric data of the patient, such as the head circumference of the patient, in order to personalize the current injected through the scalp. Okay, and this is done also with some artificial intelligence algorithms. And this is an ongoing clinical trial that we do at home in Rome. We have 20 patients enrolled currently, and we expect to have 30. So we will get the results next year of this trial. This trial is very nice because also the patients get stimulated at home as well. So they don't need to go to the clinic. They just take the device and go home for 2 months. And every day they just do this stimulation protocol. And that's it. I just want to say that this is not the work that I do by myself alone. We are a big, big team. And yeah, and as Aureli was saying, the questions, we will answer them by the end of the webinar. Thank you very much. And I leave the floor to Nacho.
Great. So thank you, Rosé. I'm Nacho. I work in the customer support department at Neuroelectrics. I'm also a part-time professor at electronic systems in Universitat Politècnica de Catalunya. And well, in this part of the presentation, we are going to see a demo of the device. Actually, we're going to see how a protocol is created. And it's a protocol that's going to be EEG and stimulation. So we will see both brain activity data capture and stimulation of the brain in one single protocol. Right, so the first thing that we need to do is to go to our software, NIC, and here we can connect to the device and create the protocol. That's what we are gonna do. Protocol editor, and we're gonna add a new protocol. First, we're gonna give a name. Usually it goes by the subject ID. In this case, I hope our CEO, Anna Mike, is watching. And as you see on the left, we have this design panel where we can select EEG or stimulation, right? And it's quite simple. The only thing that we need to do is to drag and drop the electrodes that we want to record EEG in this case. And this is gonna be the first step of our protocol. Then we can add a second step, and this step, what we'll do is to stimulate on those same electrodes. I mean, the combinations are endless. You can select different electrodes, the same electrodes, as many steps as you want. And as you see, we have many fields to select the stimulation parameters. These parameters are the ones that come from what Rosé has been talking about, the neurotwins. So each of these parameters is optimized and personalized for the patient. And now I'm going to use one that's actually from one of our clinical trials from Alzheimer's. This kind of protocol, as you see, is alternating current stimulation, TACS, that is for transcranial. And protocols like this have shown to improve cognition in healthy subjects, and what we are trying to prove is if it also helps in patients that have Alzheimer's. So I think everything's ready. We just gonna give a name to this second step and finish and save. Okay, so if we go to this step that is stimulating, we can actually see stimulation preview. So this is kind of a simulation on a standard brain model. We can see the white matter, the gray matter, how the current that we are injecting to the patient will behave, and the electric fields, right? You see it flashing because it's alternating current. If we were using TDCS, which is direct current, for instance, used in epilepsy, where you want to shut down or reduce the activity on a specific brain area that's causing actually the seizures, it will be like constant, right? Constant current. Okay, so we have this setup guide. And actually, I'm gonna do the montage on Rude. So we're gonna record the brain activity of Rude. You can put the head count.
Yes.
So you see that I know where each of the cables go, right? Each of these cables that I'll label with the numbers, and I know the position. Again, these protocols come from an optimization that's personalized. To do is to connect each of the cables, but before, of course, to create a conductive path, I need to add some gel to each of the electrodes. Actually, I'm gonna put a video so you can see it better. These electrodes from Neuroelectrics allow you to clearly see the scalp and ensure that you are putting the gel where it has to be. That's in the scalp, not in the hair, right? So that the conductive path is created properly. And now it's just clipping each of the cables to the position. So you see the montage is very easy. That's why we can, I mean, patients can do it at home by themselves or being held by a caregiver. So now we are going to connect the device wirelessly, so the patient doesn't have to be connected to a computer through a cable.
So I can run now, right?
You can run exactly. Now let's load the protocol. So this is your brain actually. This is reserve brain activity. We can check the impedance, and we can go actually to this live view panel where we can actually see the EEG representation in a head model. If we use more positions, we could see actually in a brain model. Down there, you can see the different band powers. So these are the different brain waves. And if we go back to the EEG, we can start recording, and now the data is actually being stored in my laptop. I can process it. Actually, there are some programs that can send markers to this software. I stopped it because that step was the one from stimulation.
So
We're
I'm not going to smart
we're not going stimulation. Maybe you can take out the headset now. And as you see, the montage is very easy, and the versatility, right, to record the EEG and to stimulate. So that was the demo. These are the devices that we use. I mean, usually these devices specifically, you see, it's just a hair cap. It has 8 channels. Usually in the stimulation, 8 channels is enough for epilepsy or even Alzheimer's. But we have a model up to 32 channels that can record data from these 32 channels and also stimulate. So I think now that we're done, we can start with the questions.
Okay, we can, with the Q&A sessions, you can write down them in the Q&A sections in the bottom part. The first question is by Ivan Barra. I suppose it's a question for everybody of us. It says, what data do you use and how do you extract that data? In the case of the first part of the webinar that you have seen, we are using mostly EEG data, although we can use as well data from wearables to extract the digital biomarkers. And we extract them either from EEG devices as the ones that Nacho was presenting, or any other types of devices or wearables that could give some information and insight on the behavior of the users. I don't know, Jose, if you want to comment on which data you use. And
okay, I am not mute. Do you hear me? Yes, right?
yes.
Okay. Yeah, so for different pipelines, we use different type of data. So for the more complex ones, we use a lot of data from the patient. So like the fusion MRI that I was telling you, to know how the network of the brain is actually encoded for different patients. The magnetic resonance only, so to know the structure of the brain. We can also use functional magnetic resonance to know the blood flow through the brain in order to personalize these neurotwins. But the cool thing is that we can also go very simple. So we can also work with template brains and just the head circumference. Or what we can do as well is the group optimized montages. So we know if you're a female and how old are you and which is your disease. So if you have Alzheimer's, let's say, we have a group of Alzheimer's participants and we just optimize the stimulation for each of them so that we can tailor a bit more the optimal stimulation. So we can work from many ranges of data.
Thank you, Rosette. An anonymous attendee is asking, does your company have the vision to be able to be a pioneer in preventing, diagnosing, or treating mental diseases, such as ADHD, depression, post-traumatic stress disorder, and so on? Yeah, actually, this is the vision of the company. So both to characterize this kind of diseases, both mental health as well as neurological diseases, and treating them either as a standalone solution or as a combination with other treatments. So this is depending on which disease and how hard it is to treat it. I don't know if Jose wants to add anything.
I think that's perfect, what you said. This is the main vision that we have. To cure is a very different thing. I don't think we are able right now to cure any disease, or is at least not proven yet. Hopefully we will prove it soon. But for treating the main symptoms of it, it can drastically help. So this is actually what we're aiming for.
Thank you, Jose. There is a question by Alan Diaz. Says, fantastic talk, congratulations. Thank you. What data formats are you using to handle the EEG data and why? So we can take different types of formats. The devices of Neuroelectrics use EASY, which is a proprietary data format, but can also record the standard EDF. And this is the one that we are mostly using in our works, and is the most handy and the most established one. So the reason basically is because several of the toolkits that you can use to process this data, like EEG Lab in MATLAB or MNE in Python, are compatible with this format. So you would facilitate your life a lot on that. The next question is by Federico Cavallini. Thank you for the presentation. It's been extremely interesting. About the stimulation, have you found any resistance phenomenon, a loss of efficacy of the treatment in time? That's a question for Jose. Or for...
Yes. Yeah, actually we have. So as I said, the brain is plastic, and it's like going to the gym, right? The stimulation. So if you do it every day, it will have longer effects rather than if you do it just once. And there are papers out there, for example, that show that with three stimulations in 3 different days consecutive of 20 minutes, you can have in epilepsy after effects up to 1 month. But if you do it 1 full week, so 5 times a week, 20 minutes, you can last 3 months actually, average across, I don't know how many patients were there in the study. So we hope that it's like this, so the more you stimulate the brain, the more it lasts, basically. And there are also people that they do not react to this treatment, the same as drugs, basically, but hopefully we can improve that with the modelings that we're developing.
Thank you, Rosette. Another question for you, Ferran Craven Bartel. In the optimized treatment outcomes, you show an epilepsy reduction graph with an average of 47%, but there was quite a lot of variability among patients. What do you think is the cause accounting for this variability? Could it be the type of epilepsy, or do you work always with the same type of epilepsy, for instance, temporal lobe epilepsy?
Thank you for this question. So the epilepsy type has an effect. Right now, we just work with focal epilepsies. Focal actually it's a term that is getting old because we know that epilepsy is a network thing, it's not just a focus. So, and inside these focal epilepsies, again, there are many different types that right now we don't distinguish so much, but there are malformations of the brain that maybe can lead to less effect of the electric field in them, just because they are not oriented in the way that they have to be by lesions or whatsoever. But what I wanted to tell you is this 47% is based on the fact that we thought it was a focal condition, basically. Now we want to improve it, so it is a network effect. So if you stimulate in one area, it will have an impact in different other areas. And maybe just taking into account the focus that it's with this malformation, maybe you cannot get the desired results. Right now we aim to improve and take into account this network, these networks of the brain, take the most out of them and optimize having this knowledge of these patients and see if this variability is very much reduced, and we can even get all the patients respond as much as possible, basically. So thank you for the question.
There is another question by Vlad Greco. You've been able to raise 17.5 million euros in 2021. This clearly shows that there are people who believe in your technology. However, what are your current challenges and risks, and what keeps you up at night? And I can answer from the point of view of the EEG markers. Basically, the current challenge is to get EEG spread. Not only EEG has been traditionally used specifically for epilepsy research and for sleep research. And our challenge is to really make the point that this is a useful technology for other diseases besides epilepsy and sleep, as I was showing in different cases. So either ADHD, where the FDA has already certified a solution based on EEG for diagnosis, as in other diseases, neurological or mental health diseases, where it can be useful both for the diagnosis as well as for the stratification of clinical populations. And I don't know, Rosette, do you want to add something about the simulation side of the business?
Yes, of course. So there are many challenges and different scales. So from the scientific perspective, we are working with the brain, which is an organ that is a black box right now. So a lot of research is just trying to understand how it works. So in the scientific perspective, it's always a challenge to know what actually is causing a pathology and what is different between one patient and another. Also, there are subjective experiences in depression or in pain that you don't even know how to quantify those yet in the scientific world. So this is a major challenge that we're facing. From the scientific perspective, also is the amount of data that we can gather from the patient, which is not so much and is very expensive. obviously. So the more data we have, the better models we can have of the brain of one patient, or even a better understanding of the mechanisms that are behind the pathology. So the amount of data is also a challenge that we're facing. And just to say another one that is not scientifically based, basically, is that the regulatory aspect of certifying a device is a very tough journey. And yeah, having all the documentation required and being able to enroll and to convince all the doctors to be on board for your clinical trials and to enroll patients and getting the numbers and showing that it actually works, not just in a paper, on scientific paper, but actually in a big clinical trial, is very hard, and it's a lot of work. And if we would make this process so agile so we can already get to the patient now, because we really know that this can work in epilepsy. But to regulate it is very hard. So it's like, okay, let's fight for having this regulation to convince people to trust in us and to get there. So I would say that that is one that really makes us be awake at nights, basically. Thank you for the question.
There is another question by Rodrigo Gajardo. It asks, I suppose, about the stimulation. Could stimulation be used in combination with a given drug to see the effect in each individual? Which could be the potential in this sense? I suppose it asks about the usage of so-called electroceuticals, which is the combination of electrical stimulation and drug treatments that I think it has been presented in some papers in the literature, but it's not so extensively used.
No, and right now actually we're exploring how, for example, neuroplastogens are called, that are drugs that enhance the plasticity in the brain, that could be used in combination with these devices. So we are actually trying to get some more funds to see how we can combine different drugs to make the effects of stimulation last longer. In the case of depression, in this case.
This is a question for Nacho from Alan Diaz. Epilepsy characterization with EEG is done over a period of several days, weeks, months. Have you checked if your system is resilient over long periods of time, considering its wireless operability? Thank you.
Actually, our devices have an SD card slot that you can put there, and it can record in what is known as Holter mode. The challenge here is that if we are comparing it to an ECG Holter mode, couple of electrodes on the chest, depending on the derivation that you put, the electrodes are there, do not move, and the patient knows that it has them. On the contrary, it's a head cap. If we are putting the subject on sleep, they can move, the electrodes can dry, so it's a bit more challenging. That's why this is done usually in hospital facilities where they can constantly monitor that. But yeah, Holter mode is an option with our devices.
And also, I suppose maybe Alan is interested in the battery lifetimes. So.
Yeah, with Holter mode, we can record for a bit more than a day.
Thank you. Sandra Ortiz is asking, what type of brain connectivity metrics do you use? And do they work in real time or offline? In the case of the EEG, where you can compute functional connectivity, you can use coherence. This is the most usual one, as well as phase-like index. We have tested other types of these two. I think you could compute them in real time with some latency, but you could have a reasonable real-time capability of computing them. Then we have tested other connectivity metrics like synchronicity likelihood. They are quite computational expensive, and in this sense, I don't think that you could use them in real time. It would be very interesting to understand which kind of application are you thinking of. I don't know, Jose, if you want to comment on the connectivity metrics that it's applied for the neurotwins.
Yes, of course. So Right now is the same as Aureli, so the most widely used that we're using with the volt signal, just to be different from EEG, we also use EEG, but just as Aureli was saying, is exactly the same. So for fMRI, we use also functional connectivity, dynamic functional connectivity as well, which all of these, obviously, for I have zero experience in computing them online. Everything is offline, whatever we do with the fMRI. And what else? Well, and there are other more complex connectivity metrics that we can analyze, but mainly these two are the ones that we are using. Then we have other features that we're interested in, but these would be the main connectivity ones.
Okay, thank you. There is a question by Patrick Reisinger. How do you handle EEG artifacts when calculating brain connectivity metrics? In the case of biomarkers, we are using different levels of artifact rejection or correction. Being the easiest one is to apply a threshold on the amplitude of the EEG. Then in the next level, you can apply ICA, where you can reconstruct parts of the signal that have been contaminated by EEG artifacts. And we are also experiencing with different toolkits that allow you to clean really very strong artifacts of EEG mobile data. There are some toolkits out there that are claiming to have this power. And the fourth lever, our most innovative aspect, is trying to use as well deep learning approaches, concretely LSTM autoencoders, to correct EEG artifacts. And we think this could be interesting from the point of view of using them in real time with the EEG devices. There is an anonymous attendee asking, what is your current plan for commercialization of both approaches, I suppose? I don't know if you want to start, Jose.
You can go, Aureli. This is not my expertise. I can say some words, but I prefer you to start.
In the fact, also for the EEG biomarkers, currently we are very much focused on commercializing it as data analysis services, for instance, that can be used in clinical trials by pharmaceutical companies that can use this EEG. In another line, which is not so much clinical, we are working with other companies like cosmetics companies. Basically, we provide here always with a business model of service provision, project by project. There is one small line where we provide EEG reporting to a clinical center here in Barcelona, where basically we receive the EEGs, and in 24 hours you can receive a report on the EEG activity that has been acquired. And this is conducted on a report based, is provided in a report based, so it's in a report basis, so it's pay-per use, basically. And about the commercialization, so far we are commercializing the commercialization of the treatment of the transcranial electrical stimulation. This is being sold to clinicians that want to apply them in their patients for the treatment of different diseases, and trying to certify them. We have certification already for neuropathic pain and stroke, and we are trying to certify the devices, as Jose was explaining, for epilepsy and for in a further pipeline in Alzheimer. And the idea would be further at the moment to commercialize them through clinicians and practitioners.
Okay, and regarding neuroelectrics, basically we have the EEG devices that can be already be sold, so they are FDA cleared, and also they have the CE mark here in Europe. But for the stimulation one, so all our clinical and commercialization, all our commercialization strategy is in the clinics, okay? So everything needs to be regulated and certified. And we are right now aiming for for certifying our stimulation devices in epilepsy. So we are working in a pivotal trial with 200 patients, basically, so we can certify those devices for epilepsy. And then smaller pilots, we conducted one with MDD, so with major depressive disorder. And we're doing right now this one with Alzheimer's disease, and we expect also to get some funds to move this to a pivotal trial and then get all these stimulation devices certified.
If there is no more questions, there is one more question by Vlad Greco. Amazing research. Thank you. Thank you very much. You should definitely apply to EIC Pathfinder. Thanks a lot and good luck. And that's actually what we are trying to with this combination of electrical stimulation on neuroplastogens that Jose was describing.
Totally. Thank you.
Alan Diaz wants a last minute question. What's the most challenging, painful EEG signal processing tasks that you are trying to solve now? All signals that are acquired in moving conditions are very challenging. So basically, this question of solving and correcting artifacts of moving people, conducting different types of activities. This is one of the most challenging tasks that you can imagine. And this could be a perfect playground as well for artificial intelligence-based algorithm to conduct this in real time. This would be a great application that we would be very glad to take into account. And we are developing it ourselves, but of course we are open also to suggestions. Thank you, Alan Díaz, gracias, excelente sesión, muchas felicidades y mucho éxito, muchas gracias por la atención. Y if there is no other comment or question, then we will close here the webinar. I would like to thank everybody for your attendance and Jose and Nacho for the excellent talks. And yeah, we hope we can see you soon at another Neuroelectrics webinar. And we would like also to thank you, BioCat, for the organization of the Innovation Week, where this webinar has been conducted. Thank you, everybody, and have a nice afternoon.
Thank you, Aureli, and thank you, everyone.